is Mari Järve, and I am working at one of the other institutes in this constantly growing building. That is the Estonian Biocentre. More precisely, I am from the Department of Evolutionary Biology, which is also, of course, affiliated with the University of Tartu. In fact, our department its name is slightly misleading. Perhaps it should say Department of Human Population Genetics, which is what you were told I would speak about. But to put it more simply, we study history. We just don't use the methods that historians and archaeologists do. We study history using the methods of population genetics. The reason why we do this, the reason we study history at all with any methods, is because humans are fascinated with our past. We want to know where we come from. And our history is not only recorded in old documents and archaeological finds, but also in our genome, which we inherit from our parents, which is passed on through generations and changes through randomly occurring mutations that could also be viewed as a record of the passing of time. And there is a copy of our genome in almost all of our cells. Some exceptions, like the red blood cells, but most of our cells have the full copy of our genome, most of which resides in the nucleus of the cell, and is divided between the 22 chromosomes that have numbers and then the sex chromosomes. And we have 2 copies of each chromosome, one inherited from either parent. Same goes with the sex chromosomes, of which the Y chromosome is only present in men. Men inherit it from their fathers. Women don't have it. Now, there is another portion of our genome that is not in the nucleus. There are organelles in the cells called mitochondria that provide the cells with energy, and they have their own genomes. Now, when Y chromosome is passed along the paternal line, mitochondrial DNA is passed from mother to children, because the ovum has a lot of mitochondria, whereas the sperm is a small cell, it has few, and during fertilization, the mitochondria of the sperm are lost. So we inherit the mitochondria of our mother, and therefore, mitochondrial DNA allows us to trace maternal ancestry. Daughters then again pass it on to the next generation. And why am I talking about mitochondrial DNA and the Y chromosome? They are the classic objects of study of our department. And this is because of the fact that whereas we have 2 copies of the rest of the genome, of every other chromosome, mitochondrial DNA and the Y chromosome do not have a sort of a partner, and that means that they do not undergo recombination. What is recombination? In each generation, most of our genome recombines. That is to say that some parts of, say, chromosome number 4, we inherit from our mother, some from our father. During the formation of sperms and ova, the homologous chromosomes, that is, say, 2 copies of chromosome number 4, they exchange randomly some parts. So the chromosome inherited from the mother and the one from the father, they sort of shuffle or mix their genetic information, which results in the fact that each of these chromosomes, so to say, has 2 parental chromosomes. And each of those parental chromosomes again has 2 parents, and that means that the number of these ancestral chromosomes grows the further we go back in time, and it's impossible to know which part of a given chromosome we have inherited from which of our ancestors. But mitochondrial DNA and the Y chromosome do not have a partner to recombine with, which means that they are passed from one generation to the next intact. They only change through random mutations, which are rather rare, and that allows us to trace ancestry throughout generations in a single line, as is shown here, and mitochondrial DNA then allows us to trace maternal ancestry, and the Y chromosome then paternal ancestry. And that is why we like these two parts of the genome. Now, this figure resembles a sort of a family tree. However, at the tips of these branches, we do not have single individuals, we have groups of individuals. You see, the mutations that accumulate in mitochondrial DNA and the Y chromosome, they have been accumulating over a long stretch of time, and the specific set of mutations that an individual has puts that individual into one of these haplogroups, as they are called, basically the branches of these family trees. This is a Y chromosome haplogroup tree. You could get a rather similar tree for the mitochondrial DNA, that in this group, say here in haplogroup L, we have men who have all of these mutations starting from here in the root. The mutations are marked on the branches, and there are a lot of people who have this set of mutations in common, and these belong to haplogroup L, whereas these others belong to haplogroup E or whichever other letter. And the haplogroups, the branches that are closer to the root, are more ancient. These A and B, for instance, in case of the Y chromosome, are found only in Africa. And these here are then more recent, have arisen recently because, as I said, that there is no recombination. Mitochondrial DNA and Y chromosome only change through accumulating mutations. And obviously, the more recent ones then have had time to accumulate more of these mutations. Now, this I just wanted to explain so that the word haplogroup would not be terribly unclear as it was for me when I was a bachelor student at first. But generally, the spread of any one haplogroup, whether it is a haplogroup of mitochondrial DNA or the Y chromosome, is limited to one continent, because when the haplogroup arose, when the mutations that define it arose, then the people carrying these mutations have not spread very far. They are usually limited to one continent. These different colors here show the frequency of different haplogroups. Okay, but this is not always the case. And one such example is the Y chromosome haplogroup R1a, a rather recent haplogroup. This probably arose less than 10,000 years ago. And the men carrying the mutations that define this haplogroup are spread, as you can see, rather widely. This haplogroup is frequent in Eastern Europe, but it is also frequent in India and Central Asia, not so frequent in between. But this spread is not typical. And this is why this haplogroup has been an object of study, because it is not likely that the carriers of R1a in Europe and in India are entirely alike. They have the similar set of mutations. They are in one haplogroup, but there are probably some mutations that we haven't discovered yet that divide this spread, so that these men and these men are somewhat different from one another, that they belong to, so to say, subhaplogroups or subsets of R1a. R1a in the general tree is positioned here. If we zoom into haplogroup R, these lineages belong to R1a, and basically now you can see that there is also structure within each haplogroup, that there are these sub-haplogroups, branches. They all have a long set of mutations in common, starting from the root. They have all of these mutations and all of these, but then they have some that differentiate them from one another. But so far, although we know here, how many? 7 different sub-branches within R1a, we didn't know anything that would differentiate this spread. We didn't know any branch that would be, say, present only in Europe or only in India and would allow us to see what the difference is between these two groups and how they might have spread. But finally, the first breakthrough in this was when a group involving our department and many collaborators from abroad discovered the marker that defines a European-specific branch within haplogroup R1a. And this mutation is called M458, and it is present only in the R1a carriers of Europe, of Eastern Europe, and it is rather frequent, most frequent in Poland and also here in Russia and in the Ukraine. And this then defines this part of the general spread. And also, interestingly, the areas where this sub-haplogroup, R1a M458, is frequent, correlate rather nicely with areas where several Neolithic cultures were spread around 8,000 years ago in Europe, and such as the linear pottery culture. And also, it is possible to estimate the date of the spread of a haplogroup based on genetic data. And this also matches the time about 8,000 years ago that this haplogroup began to spread in Europe, and also that these cultures spread, as we know from archaeological records. So it is likely that this sub-haplogroup, or R1a, spread and achieved such high frequency in Europe due to the fact that it was carried along with this culture, that the population size rose, and this haplogroup spread then reached rather high frequency. Now, so far, I've talked about the Y-chromosome, mostly, also mentioned mitochondrial DNA, but as you have already heard, these are only a small part of the picture. There is a lot of information we are missing, and the question is, can we do anything with all the rest of it? As I told you, recombination that occurs in most of the genome makes it impossible to trace ancestry based on that information, but otherwise we can use information from the whole genome to determine whether two individuals are similar or not. And this is then, as Professor Andres Metspalu already talked about, for instance, done with gathering information with these chips from hundreds of thousands of genetic markers across the whole genome. If we study these hundreds of thousands of markers in many individuals, we can see that some of them are more similar and some are dissimilar, and this usually follows geography. That is to say, people are similar if they live close to one another, and dissimilar if they are far from one another. And it has been shown repeatedly, also by just now in the last talk, and in many papers, that these genetic plots match geographic maps very well. This is a rather famous figure from a study already 5 years old that shows how well this matches European geography. These are European populations, several 100,000 markers studied in each of these individuals, denoted by these small abbreviations, and colored according to their nationality. And these colored circles represent the average of each population. And you can see that if you turn the genetic plot the right way, I mean, considering north and south and east and west, then you get a plot that matches geography very well. And in fact, this has become sort of the null hypothesis or the thing that we usually see. When it gets interesting when there is something that differs from that usual geographic spread. One example of the use of these hundreds of thousands of markers, basically point mutations, one nucleotide change across the genome, is a study from our lab and collaborators from abroad that focused on the Caucasus. The Caucasus is a mountainous region between the Black and Caspian Seas, and it has very high ethnic and linguistic diversity. Now, another point we could make is that this already shows that linguistic data does not usually match genetic data. We get the picture we get from geography, that is that all these populations from the Caucasus reside quite closely as they do on the map. But linguistically, they are very different from one another. The Georgians come from a whole different language family. There are 3 indigenous language families in the Caucasus, plus Indo-European and Turkic languages spoken, and if this followed linguistics, then they should be on probably separate sides of the plot, but they're not. They're geographically close, and genetics matches that. And it is also logical that they are close to the Near East, from where the Caucasus was probably populated. But it is strange that there is a rather wide gap between the populations of North Caucasus and Eastern European populations that live quite adjacent to them, Russians, Ukrainians, but they're different. And this sort of a finding based on these markers across the genome prompted us to look at this difference in more detail. And we found that, in fact, there has been sort of a barrier to gene flow, that there has been practically no gene flow from the Caucasus to Eastern Europe, possibly because there was once a water barrier between the Black and the Caspian Seas about 12 to 14,000 years ago. This could be one of the reasons. There could be others. But this sort of a general picture points you to the things that are interesting, that are worth study. Now, this is still based not on the whole genome, but on several 100,000 positions across the genome. The future is actual whole genome sequencing, that is determining the nucleotide sequence of most of these 3 billion base pairs in the human genome. As you know, this has already been done for the human reference genome and so on, but now more and more studies are doing this for individual genomes, for hundreds, thousands of individuals, to compare them to one another. And you see, we cannot sequence all of these 3 billion base pairs in one go, in one reaction. It would be great if we could, but we can't. And we sequence these small fragments, these stretches of the genome that we then have to map according to the reference genome sequence. That we have to know how they're positioned here. And as you can see that these small stretches, they overlap. And in fact, in order to be sure that we have sequenced some nucleotide correctly, that we know whether it's A, T, G, or C, we have to sequence it repeatedly. This is what it means that we have to sequence at high coverage. That basically there have to be a lot of these small stretches sequenced that cover this one position, and then we can be sure that it is not a sequencing error, or well, relatively sure anyway, that it is not a sequencing error, that we actually see what nucleotide is in any given position. Okay, and again, Professor Metspalu already talked about this. We now have these so-called second-generation sequencing technologies, and we're hoping for even better ones. This is a field that develops very quickly, and there are companies that offer this service of sequencing whole genomes, such as Complete Genomics in California, that was recently purchased by the Chinese giant BGI, and then also Illumina. And we at the Estonian Biocentre and our lab have also ordered the sequencing of hundreds of genomes from the company Complete Genomics. Now, these numbers on this slide have changed slightly because by now we have already received all the genomes from the first batch, that was 284 samples, and we have also sent some additional ones to them for sequencing. And as you can see from this table, most of them have passed quality controls, and the data can be used. And this map shows the locations of the samples. We adopted the strategy that we would try to cover many populations, but only 2 to 3 individuals from each population, because although this has become considerably cheaper, it is still not what you would call cheap. So we have to set some limits. Obviously, there is much more information in the whole genome, and this data is now being analyzed, and this is where our field is going, sort of. Not just the Y chromosome and mitochondrial DNA anymore, but also comparing whole genomes. Now, I think that the first title of my talk that you were given was Population Genetics in Estonia. That's true, but it's not only in Estonia, or at least it's not conducted only by Estonians, our research. This is our group, or most of it, led by Professor Richard Villems. And we have collaborators from Israel, India, Belarus, Bosnia, Armenia, Russia. So I have always felt that I don't really need to go abroad to do international research, because our group is we once had a sort of a party, Valentine's Day, I think, and we counted how many nationalities we had around the table. I think we got 9 at the time. And thanks are owed to all of them. And with a picture of the Caucasus that I was talking about, I thank you for your attention, and I'll be glad to answer any questions.